In May 2026, the journal Cell Metabolism published research showing how a molecule derived from garlic influences the biological process of muscle aging. The study focuses on sarcopenia, the involuntary loss of muscle mass and function that typically begins when a person reaches their 30s. This chart tracks the trajectory of that decline. Without intervention, many individuals lose 50% of their muscle mass by age 80, reaching a state of physical frailty. While resistance training is the standard defense, these new findings suggest that muscle health also depends on a communication system that connects multiple organs throughout the body. A research team from the Institute for Research on Productive Aging in Japan investigated the clinical properties of aged garlic extract to understand its effects on physical vitality. Within the extract, they identified a specific bioactive sulfur compound known as S1-propenyl L-cysteine, or S1-PC. The researchers observed that S1-PC does not interact directly with skeletal muscle fibers. Instead, the molecule initiates a biological sequence that begins in an unexpected location. After ingestion, S1-PC travels to white adipose tissue, or fat cells. It binds to and activates a metabolic regulator inside the fat called the LKB1 enzyme. This triggers the cells to release extracellular vesicles into the bloodstream, carrying a longevity enzyme called ENAMPT. This process shows fat tissue serves as a primary dispatcher in the body's aging signaling network. The ENAMPT enzyme travels through the blood to the hypothalamus, the brain's regulatory center, highlighted here in blue. Once there, ENAMPT increases the production of NAD+, a molecule used for DNA repair and cellular energy. This increase in NAD+, prompts the hypothalamus to send signals through the sympathetic nervous system toward the skeletal muscles. This nervous system signal commands the muscles to restore their youthful levels of force and contractility. It forms a three-way biological relay. Fat tissue signals the brain, triggering the muscle to restore strength. These charts from the cell metabolism study show the results of long-term S1-PC administration in aged mice. The mice showed reduced frailty scores, restored temperatures, and increased muscle force, corresponding to the dense green cell structures in the upper panel. In human trials, a single dose of S1-PC increased circulating ENAMPT levels in middle-aged subjects within 2 hours. Because this fat-brain-muscle pathway is conserved across species, S1-PC represents a documented intervention for age-related decline. This breakthrough research indicates that maintaining physical function through aging requires supporting the body's internal communication network. S1-PC-based nutraceuticals provide a safe, side-effect-free method to keep the signaling pathway active. Preserving this relay is a documented factor in maintaining physical independence well into the golden years. To stay updated on the science of healthy aging,